Hi everyone, welcome to this session. And first of all, we'd like to thank CTF to organize this meeting and also all of the speakers to share their unpublished work with us. So yesterday we saw different approach to treat the tumors by drug combination, immunotherapy, and also vaccination. And in this session, we are focused more in the different improvements to restore and repair the disease-causing mutations and avoid the development of neurofibromatosis. So it's a pleasure to me to introduce Dr. Zhou, that is an associate professor of neurosurgery and biomedical engineering at Yale University. His research centers on development of nanotechnology approaches for drug delivery to the brain and for non-viral gene delivery, and applying them for treatment of various diseases, including neurofibromatosis. So, please, Dr. Zhou.
Good morning, everyone. My name is Jonathan Zhou. I'm an associate professor at Yale University. It's a great pleasure to be here to talk about our recent progress in development of non-viral gene therapy for neurofibromatosis. So in today's talk, I will first give some background information about gene therapy, including the status of gene therapy and delivery as a major hurdle for translation of gene therapy. Next, I will talk about the development of polymeric nanoparticles for gene delivery in my lab. And lastly, I will talk about our recent progress in collaborative evaluation of polymeric nanoparticles for gene therapy. So first, gene therapy. What's gene therapy? So gene therapy is a technique for correcting defective genes responsible for disease development. So there are three major types of gene therapy. The first type called gene augmentation or gene replacement therapy. In this case, a copy of function gene is delivered to target cells or tissue to enable restoration of the function gene, the function of the specific gene. So the second type is called gene suppression. So in this case, either siRNA or shRNA or mRNA is delivered to target cells to enable target lockdown of the specific gene at the translational level. So the third type of gene therapy is called gene editing. So typically, a nucleus is delivered to target cells. So this nucleus can initiate either gene correction or lock down or lock in a specific gene. So gene therapy actually is not a new concept, has a pretty long history. So first gene therapy clinical trial was approved in 1989. And now, every year, there are more than 300 clinical trials worldwide. However, development of gene therapy hasn't been easy. In particular, there's a tragedy happened just right after the approval of the first gene therapy. In 1990, a boy received gene therapy in which adenovirus was used to deliver a gene called OTC. Unfortunately, infusion of adenovirus kills the boy. So this tragedy really reminds us that the gene therapy could not be safe, particularly when viral vectors are used. So this slide summarizes all the 22 gene therapy products which have been approved worldwide. And in the US, the first gene therapy product was not approved until 2017. So mainly because of all the safety concerns. So among the 22 among the approved gene therapy products, majority of them were designed for treatment of monogenetic diseases such as M1 or M2 disease. So among the 22 gene therapy products, you may find that most of them are using viral vectors, because some of them use vector DNA or plasmid DNA. But in reality, is that this type of gene therapy always suffer from very limited efficiency. So those viral vectors are used mainly because they're able to deliver genes or genetic materials in high efficiency. So as shown here, those viruses somehow develop a mechanism which allows them to penetrate cells and release DNA in high efficiency. However, it's also known that using viral vectors is associated with a lot of problems. Now, for example, viral vectors can potentially induce illicit immunogenicity, and it can cause insertion mutagenesis. And also, it's not easy to produce viral vectors, and also viral vectors are not easy to modify. Related to ANF disease, so viral vectors may not be the best choice, particularly for ANF1. So ANF1 cDNA is huge; it's over 8 kilobase pair. So none of the existing viral vectors alone, a single vector, can deliver such a large gene. Do not have this degree of capacity. So another concern is that for many ANF patients, probably they need to receive gene therapy multiple times throughout their life. So in this case, viral vectors could be used in the first few treatments, but with time, the body will develop an immune response and eliminate subsequent viral injection of virus. So basically, the gene viral-based therapy could be effective in the first few cycles, but with time, the virus therapy will lose effect, that is eliminated by the immune system. So therefore, ideally for ANF disease, non-viral vector could be the best choice. So the many non-viral vectors have been developed. So by far, the most efficient vectors should be synthetic nanoparticles. So what's a nanoparticle? So nanoparticles, by definition, are devices inside our nanosite, typically about 50 to a couple hundred nanometers. So another way to understand the synthetic nanoparticles for gene delivery is that these nanoparticles are inside our virus. So basically, synthetic virus. So the advantage of non-viral vectors is that these vectors typically are safe. They are all made of synthetic materials. They're easy to produce. They are flexible for modification. And another major advantage is that non-viral vectors are known for delivering our large payload, like ANF1 cDNA. So there's a disadvantage, of course. So traditionally, non-viral vectors are associated with low efficiency. Fortunately, a lot of progress has been made just over the past few years. So now, many vectors can deliver genes in pretty good efficiency. Now, for example, this year, I believe many of the audience have received COVID-19 vaccine. Probably know that both the Pfizer vaccine and the Moderna vaccine are delivered using lipid nanoparticles. So this particle can deliver mRNA in pretty good efficiency. So in my lab, we mainly focus on polymeric nanoparticles for gene delivery. So the development of polymeric nanomaterials for gene delivery actually has pretty long history and can be traced back to 1960s. So when the first generation polymers was developed, these polymers including PLL, PEI, both are very classical gene delivery polymers. So one major feature of these polymers is that there's a lot of amino group on the polymer chain. So this amino group provide charge, positive charge. As a result, these polymers can condense negative charge DNA into nanoparticles, can interact with cells and get into cells. As a result, we can deliver a genetic payload into cells. So the polymer charge is very important in terms of condensing DNA and penetrating into cells. However, since there's a large amount of polymer charge, it also causes problems. So this charge will induce significant toxicity. So therefore, the feature of first generation polymers, typically they have moderate or significant toxicity. They can deliver gene in good efficiency, cannot say very high, but typically they cannot deliver gene well in vivo because they have limited stability. To overcome those limitations, we recently developed a family of low polymers, low polymers. Two polymers by definition, these polymers are made of three monomers, including lactone group, DES, and MDEA. So those polymers are unique because within a single polymer molecule, we can tune three major parameters, including charge, which is provided by MDEA group, and the molecular weight, which can be controlled through the polymer reaction time, and hydrophobicity, which is provided by the lactone group. So what we found is that the gene delivery ability of polymers actually not only determined by charge, but actually determined by all three major parameters. So as a result, we found those polymers can condense DNA into nanoparticles and they can deliver gene in high efficiency. So in particular, because we use a limited amount of charge, positive charge, those particles are very safe without induce significant toxicity either in vitro or in vivo. So this is the first generation polymer we have synthesized within this library. Some of them are liquid polymer, some of them are solid polymers. We have fully characterized all those polymers. We found that within this family, this liquid polymer can deliver gene very well. For example, I show here, there's a commercial agent, lipofectamine. Compared to the leading commercial agents, these liquid particles can deliver gene in efficiency typically about 10 to 50 times greater. However, we realize there's a limitation with the liquid particles. So although they form particles in solution, these particles do not have the stability and collapse after lyophilization. Of course, we want to synthesize solvent particles, right? So it's easy for clinical translation, and also there's sufficient stability for in vivo use. So unfortunately, the solvent polymer in this family, which also form solvent particles, cannot deliver gene in good efficiency, much less than commercial agents. So recently, we started to develop a second generation polymers with a goal to overcome all the known limitations. So what we have done is that we significantly modify the chemistry through incorporation of function molecules, such as this molecule to enhance target delivery, to enhance affinity with DNA, to enable conjugation of cell conjugation peptide, and to enable the polymers to respond to stimuli. So by far, we have synthesized over 300 polymers, and while our synthesis and characterization is still continuing, we have identified several lead polymers which can deliver gene very well. So one example is shown here. So this is the lead polymer we have synthesized. So the polymer responds to stimuli within cells. So what we found is that this polymer form solid nanoparticles that show here with very distinct sphere shape. So these particles can deliver gene into cells very well. And right getting into cells, these particles collect and release genetic payload. So as a result, we found that these particles can deliver gene in very high efficiency compared to commercial agents. Those solid particles can deliver gene efficiency probably 20 times greater. So we further evaluated these particles for delivery of gene editing. Here we use traffic-like reporter cells. So basically, those cells do not repress red fluorescent protein at normal condition. Only if the gene editing of CRISPR-Cas9 is successfully delivered, the CRISPR will initiate cut here. So therefore, to sort of chance the cells that express RFP. So we test our polymers for delivery of CRISPR-Cas9. So the results are shown here. So basically what we found is that our particles can deliver CRISPR-Cas9 in extremely high efficiency compared to commercial agents, which in which only probably 5% of cells express RLP. Our particles enable probably over 60% of cells express RLP. So those findings were further validated by confocal imaging and also the survival assay. We also evaluated this particle in vivo. So in this case, we inject particles into TLR transgenic mice. We inject particles into the brain through either local delivery injector, local convection enhanced delivery, or RCV injection. So in both cases, we found that our particles can edit cells within the brain. So with those particles, recently we start to characterize our particles for NF1 gene therapy. So we are fortunately to have the opportunity to collaborate with Professor Castelson, a leader scientist in this field who has developed a variety of NF1 genetic models. So in the first one, we use acute model. So for this transgenic mice, when tamoxifen inject, tamoxifen can initiate acute NF loss, so which confirmed here. So what we found is that acute NF1 loss can induce quick loss in body weight and body fat. As a result, this mice died just in a few days, very fast. So using these mice, we first test if our particles can deliver NF1 cDNA into brain cells. So we inject particles to the brain through RCV injection. And then we isolate brain tissue and perform Western blot analysis. So the results are shown here. So we found that the single RCV injection significantly enhanced NF1 expression in the brain. So then we test if the RCV injection of particles can enhance the survival of the transgenic mice. So in this case, we inject particles into the brain, and 2 days later, we inject tamoxifen to induce NF1 loss. So what we found is that in the treatment group, a fraction of them, only 20 to 30% of mice survive a long time, never die. So this is quite interesting because in the control group, all mice died within a few days. Of course, this is a pilot study. We believe the efficacy can be further improved because the NF1 loss, not just in the brain, actually in all the body, throughout the body. So one can imagine if we deliver NF1 intravenously, and the particle may be stored NF1 in peripheral tissue, right? So in that case, I'm pretty sure that restoration of NF1 in the organ other than brain also provides survival benefit. And also we can also inject particles multiple times into brain to enhance NF1 expression. So this work is still ongoing. So in the second study, we test our particles in a transgenic rat model. So this model is quite unique. So this rat started to develop breast tumors after sexual maturity. So using this rat, we first test if our particles can deliver genes into the breast. So here we inject particles into the fat pad, and then we isolate the breast and imaging the breast. So as shown here, we found that single injection particles enable expression of RFP protein. So more recently, we started to test if the NF1 loaded particles can provide any therapeutic benefit after injection. So the result are not shown here. So mainly because really that's too exciting, and we want to really validate the finding before we can present results to the community. So basically, what we found is that the single injection particles really can prevent tumor development. And those not only prevent development, we also found that single injection particles actually can regress established tumors. Again, as I said, this is a pilot study. This is a very preliminary result. Right now, we repeat this study using multiple animals. Hopefully, we can share the exciting results with the community in the next few weeks or months. We also start to test our particles for delivery gene editing or treatment. And this is the ongoing efforts. So summary, gene therapy is a promising approach for treatment of various diseases. And the delivery represents a major hurdle for clinical translation. And we have developed a new generation polymers which enable efficient delivery of gene therapy. And we have demonstrated that delivery of gene therapy or gene editing therapy via our particles might enable efficient treatment of infectious disease. Of course, you may ask, the animal result looks good. What if the result can further validate? Conditional therapy can be translated into human patients, right? So here I want to briefly talk about that. So I will not compare our particles with virus. So I would like to compare our delivery system with a delivery system which is used for COVID-19 vaccine delivery. So as far as I know, our particles can deliver gene efficiency at least comparable to the lipid particles. And also in terms of production, our particles can be produced with a high throughput microfluidic device, so which is at least comparable to the lipid particle production. When compared to lipid particles, our particles have many advantages which make them suitable for translation. So our particles made of 1 to 2 components. Right now, our particles made of 2 components that try to change the chemistry. So hopefully we can make 1 component particle. So in terms of toxicity, our material is fully biodegradable, which means that the particle will be dissolved in the body automatically. So toxicity-wise, much lower compared to lipid particles. Only because of degradation, this particle can release payload, genetic payloads over time to allow for production of persistent gene transduction. And our particles have high degree of stability. We produce a particle in solid powder, which are stable even at room temperature for many days. So therefore, it's possible, it's quite promising, or at least can say that. So our nanoparticle-based gene therapy could be used for treatment of inflamed patients. Acknowledgement, finally, I would like to thank the members in my lab, current members or previous members. I would like to thank my collaborator, Professor Dr. Casperson, and his lab. All the calculation, circuit evaluation work was done in his lab. Finally, I would like to thank my funding support agency, including Gilbert Foundation and the NIH, particularly the ECGE consortium. Thank all of you for your attention. I'll be happy to take any questions. Thank you.
So thank you, Dr. Zhou. If there are any question in the audience, please use the chat. Yeah, there are one. So does the vehicle also reach the brain? And it does. It penetrates through the central eye, the CTF brain barrier or blood-brain barrier.
Sorry, can you repeat this question again?
Yeah, you can see in the chat, but I repeat it. So, Abedo Achiji asked if the vehicle also reaches the brain, and if it could penetrate the brain barrier or the blood and the blood barrier. So if one injected, it could also transpass the CCF barrier.
So mainly about the particles cross the blood-brain barrier.
Exactly.
So that's a very good question. I think that for anyone knows me, probably you may know that my lab focus on two major things. One is gene delivery, one is engineered particles for crossing blood-brain barrier. So the current particles are not engineered for crossing the BBB. So that's why we choose a CV injection for now. But actually, there's our next step, of course, to engineer particles for crossing the blood-brain barrier. So this is an ongoing work. Hopefully, we can achieve that.
So we're waiting for more questions from the audience. I have one. So regarding the way it is packaged, the CDNA from NF1, it should be protected in any way to be stable inside the particle for long term.
The particle for long term, right? Long-term effect, right? So yeah, I think as I mentioned, there's probably the advantage of the use of polymeric nanoparticles. Certainly, at least in vitro, we found that in other study, in vitro in vivo, we found that those particles really can provide the long-term effect. Probably due to the polymer degradation, because this is the polymer means that a long chain of molecules. The particles release payload through degradation. So basically, they just shrink or degrade with time. So the payload will release over a certain time. So why it's good for persistent gene delivery.
Okay, thank you. So if there are no more questions, no. Maybe we can open the discussion after all the talks. So yeah, this is the turn of Matthew Duger.
Yes, so maybe we can move to the next speaker, Dr. Mathieu Drouillet, who is a French early career researcher who moved to Australia after his PhD to fulfill his true interest in gene therapy using viral vectors. And he will share today his passion for AAV capsid development to improve therapeutics delivery.
Hi everyone, my name is Mathieu Drouillet and welcome to my presentation. First, I would like to thank the organizer for giving me the opportunity today to speak about the work that we are doing at the Children's Medical Research Institute, which is about developing novel AAV variants targeting the primary Schwann cells. I have nothing to disclose. So first, I would like you to introduce adeno-associated virus, or AAV, which is a small virus that does not cause any human diseases. It is composed of a protein capsid of around 26 nanometer of diameter and a single-stranded DNA genome. The genes are the rep and cap and are flanked by two ITRs, or inverted terminal repeats. In another hand, AAV as a vector is composed of the same capsid, but encapsulate a genome that is devoid of all the virus elements and have a therapeutic gene expression cassette instead, which can be a promoter with your gene of interest. The ITRs are the only sequence retained which are needed for packaging. The AAV capsid is the primary determinant of the vector tropism, which means it determines the tissue and cell specificity. And there is a lot of effort to design novel AAV capsid to develop the ideal gene therapy vectors. So the capsid can be engineering or modified to retarget the vector to the cell of interest and to make the delivery more efficient. So for this purpose, in our lab we generate a lot of diversified variants which can be up to billions of different variants in so-called AAV libraries. Those libraries can be made by using different methods. One of them is a DNA family shuffling, which is taking the full-length capsid of the parental AAV capsid, fragmenting them with DNAs, and reassemble them in a random manner by partial homology. Another option that we use often in our lab is a peptide insertion. So we take a very specific exposed part of the viral capsid protein and insert randomly 7 amino acids on the surface, which will at the end modify the AAV cell surface binding. With those libraries, we will apply directed evolution to identify the base candidate that is driven by selection pressure. And our target of choice is the Schwann cells that are depicted here on this picture. And in the lab we are working with the primary human Schwann cells that are, as a model, that have been purified from nerve or from skin. And if you scan literature, there is already 2 papers that identify some AAVs that are can transduce primary human Schwann cells, and especially a paper from Bay in 2019 identify AAV-DJ, which is the first chimeric capsid ever made back in 2008 by the Mark Kay's lab, which is the best at transducing a human primary Schwann cell so far. So in order to identify and develop better capsids than the AAV-DJ, we took 2 different approaches. The first one was a comprehensive screen where we tested 51 different capsids. And in order to test this 51 capsid in a fastest way and to not use a lot of human Schwann cells, we developed this multiplex comparison method based on a unique barcode identification. So each capsid will be packaged with 2 unique barcodes, which is a 6 nucleotide sequence. All the capsid will be mixed, and we'll transduce the primary Schwann cells they are from nerve or skin or fibroblast, and after 48 hours we can harvest the cells, extract DNA and RNA, and PCR amplify this barcoding region and submit it for NGS analysis. This NGS will give us 2 sets of data, one at DNA level, which corresponding to the cell entry or physical transduction, and one at RNA level, which will be the expression or functional transduction. The second method we used was the selection of novel capsid using our proprietary selection platform, which enables us to select based on transgene expression. We used 3 different libraries, 2 peptide library based on AAV2 and AAV9 capsid, and 1 DNA family shuffling library based on the capsid AAV1 to 12. So those libraries were cloned into our FT platform. We transduce the primary Schwann cells, wait 1 week, and fax sort the GFP positive cells to recover the full-length capsid for the DNA family shuffling or the peptide sequence for the peptide library, and do some iterative round of selection. So first I will present you the result that we get with the AAV kit. So at DNA level, we were able to identify a lot new variants that can transduce with better efficiencies than the benchmark AAV-DJ, which is here, and also can express better than AAV-DJ, which is again here. And those capsids that are efficient at transducing both skin and nerve-derived Schwann cells are also very efficient at transducing fibroblasts. That means they are not specific to the primary human Schwann cells, and that's why we need to develop novel AAV variants. So as I said before, we choose 3 different libraries, and with the AAV2 peptide library, we selected a top 10 peptides after 2 rounds of selection for further testing. For the AAV9 peptide library, we selected the top 12 peptides after 2 rounds of selection. And regarding the DNA family shuffling, after 5 rounds of selection, we cloned 15 candidates, and we selected at the end top 4 variants. So all of those variants were mixed together and packaged with 2 unique barcodes with variants identified from the AAV kit, and also including the AAV DJ as a control. And then were mixed in what we call the super mix. This super mix was titering, and we transduced the primary sequence cells, and after 48 hours we extracted again DNA and RNA and PCR the barcoding region and submit this to NGS analysis. And we get with these results at DNA level. As you can see, there is not a major difference between all those variants, which can enter the human sequence cells at pretty similar level, except for some variants that are depicted here. And the most interesting part is that RNA or expression, where we can find one of our novel variant, the PEP human SC1, which is the best at expression with almost 10% of the reads, which is 2 times better than all other best variants so far. And so those variants, for example, are the variant from the DNA family shuffling. And especially interesting, it's 8 times better than AAV-DJ. So we choose AAV-DJ on PEP1 for further testing. We first test this variant on fibroblasts using different MRI and measure the percentage of GFP positive cells by flow cytometry. And as you can see, with a MOI of only 1000, AAV-DJ transduce around 70% of the fibroblast, as opposed to PEP human SC1 at transduce less than 5% of fibroblast. And even with a IMY of 20,000, our novel variant transduce less than 25% of fibroblast, as opposed to AAV-DJ. So another term, PEP human SC1 has a lower tropism for fibroblast. We also perform immunocytochemistry analysis using 2 different culture of primary human sequence cells, one called PN09.2, which is a mixed population of sequence cells labeled with the marker S100, as you can see on this picture, and also a population of S100 negative, which will be fibroblasts, depicted with a white arrow. So it's a population 60-40%. The other cell culture that we used was PN02.8, which is highly pure with 90% of Schwann cells. So we used an MOI of 1000 to transduce those culture with AAV-DJ and PEP human SC1. We wait 72 hours and fix the cells and do some ICC. So as you can see with those results, our novel variant, PEP human SC1, transduce more than 70% of S100 positive cells with human Schwann cells, which is significantly higher than AAV-DJ, which has a higher efficiency and specificity for the fibroblast or S100 negative cells, which are around 70% of GFP positive cells, as opposed to our novel variant, which only transduce 25% of fibroblasts. When we use the PN02.8 cell culture, which is highly pure, again we saw that PEP human C1 transduce more than 80% of the primary human Schwann cells, again labeled with S100 in red, as opposed to DJ that transduce around 20% of the Schwann cells and even transduce 1 fibroblast depicted with this white arrow here. So overall, our results show that PEP human SC1 is the best performer in primary human Schwann cells. So in conclusion, and to this talk, we select already existing AAV variants using our AAV kit, which have higher efficiency than AAV-DJ, but have also are not specific to the primary Schwann cell. That's why we developed novel AAV variant, which show higher transduction efficiency and a lower off-target. What we would like to do now is to test transduction efficiency, but with the presence of AAV neutralizing antibodies, We also test those novel variants in vivo in a humanized mouse model, which is repopulated with primary human hepatocytes, also called human FRG model, to test the liver detargeting of those variants. And finally, in order to develop a better capsid to target the human primary Schwann cell with higher specificity and efficiency, we will design new library with higher fitness, which are based on previous AAV screens in this project. Finally, I would like to thank my team at CMI, especially Lishek, Sam, and Christy. I would like to thank also my collaborators at Calgary, Jeff, Tak, and Elodie. I would like to thank Professor Margaret Peggy Wallace. And finally, I would like to thank the CTF Foundation for their financial support of this project. Thank you.
Thank you. That was a great talk. Unfortunately, Matthew could not attend the session today to answer questions. You can, of course, send him your questions by email, and we can also forward the questions from Slido to him. And so we are going to move to the next speaker, Dr. Stephanie Joy-Bouley.
Yeah, she's a research fellow at Massachusetts General Hospital under the mentorship of Dr. James Walker. And she's the recipient of Dorothy and Spiro Lattice Fellowship in Neuroinflammatory Research from Boston Children's Hospital, and is a former CTF Young Investigation Awardee. So, please, Stephanie.
Good morning, everyone, and thank you for being here. Today, I'd like to talk to you about the potential problems and innovations associated with CRISPR-based gene therapies and the pipeline that we're trying to develop for testing corrective therapies for NF1. There are some challenges when it comes to correcting NF1 using gene therapy. We know that due to the large size of the NF1 gene, complete NF1 gene replacement therapy is not currently feasible. In addition, gene editing strategies are further complicated by the mutational complexity associated with neurofibromatosis type 1. Despite this, there are current gene editing tools available that have the potential to correct a subset of NF1 mutations, as shown here. We are currently investigating using CRISPR-based gene editing to correct NF1 mutations. One limiting factor of CRISPR-based gene editing is the PAM site. PAM sites for the wild-type Cas9 and Cas12a are shown here. For the RNA guide to target the desired DNA region, there must be a PAM site in close proximity to the target, as the RNA guide, along with the PAM site, dictate docking and cutting of the Cas enzyme. This is true for all of the gene editing strategies that we are pursuing. We are utilizing both classic CRISPR-HDR gene editing, as well as the latest CRISPR-based editing approaches, base editing and prime editing. With CRISPR-HDR, donor DNA serves as the template for repair, in addition to introduction of the Cas enzyme and the guide RNA. However, there are limitations to this approach, as it induces double-strand DNA breaks and requires an exogenous donor DNA repair template. Base editing and prime editing, on the other hand, are generally more precise and exhibit lower levels of off-target editing as they result in single-strand DNA breaks. Both of these approaches require additional enzymes, which are part of the Cas complex, which help drive the efficiency and fidelity of correction. As I mentioned previously, in all of these approaches, we are limited by the PAM sites within our region of interest, which can significantly limit the areas that we can target for correction. To address this, we have collaborated with Dr. Ben Kleinstiver at Mass General Hospital to use protein engineering to increase our targeting repertoire, which is essential for targeting NF1. Ben's work has led to an increase in PAM sites that are recognizable by Cas enzymes. Additionally, screens are being conducted to increase the efficiency and fidelity of Cas editing. These Cas variants can be applied to all of the CRISPR-based gene editing approaches that I've introduced, including base editing and prime editing. And so, in order for us to devise corrective strategies, we need a source of NF1. Ideally, this would come from a patient in the form of a blood sample, which can be used to derive immortalized lymphoblastoid cell lines with a specific germline NF1 mutation. These cells would serve as the template for our designing of corrective strategies. We can then introduce these corrective strategies using transfection and nucleofection and determine the efficiency and fidelity of these strategies using next-generation sequencing. In the absence of patient-derived cell lines, we've taken an alternative approach using immortalized human Schwann cell lines from Peggy Wallace and Amit Hoke, which we can CRISPR to introduce specific NF1 mutations of interest. These cells then serve as the template for designing our corrective strategies. As proof of principle, we've selected 3 mutations to model and correct, 2 point mutations and a small deletion. Today, I'm going to be focused on one of our point mutations. We picked a potentially recurrent point mutation, which has been identified in 4% of NF1 patients, and designed several strategies to introduce this mutation into our immortalized wild-type human Schwann cells. It's important to note that all of these strategies rely on engineered Cas variants, as they cannot be altered using wild-type PAM. We tried all of these approaches, and the one that was most successful was found to be our adenine base editing approach. We were able to successfully generate a series of cell lines where we've introduced a point mutation, indicated by the red arrows, without affecting other residues. At the top, you can see the area that served as our RNA guide, as well as our PAM site, which was an NGT PAM. Because we know this mutation results in a shorter cDNA product due to skipping a portion of exon 10B, we wanted to devise a PCR strategy shown here that would allow us to examine alternative splicing patterns. And we found that when comparing our wild-type heterozygous and homozygous cell lines, we do in fact see an altered splice pattern. RT-qPCR demonstrated reduced expression in the heterozygous and homozygous cell lines when compared to wild-type cells, both prior to and after the point mutation in the NF1 gene. And increased growth was observed with the homozygous mutation compared to our wild-type and heterozygous cell lines. By taking advantage of the engineered and expanded PAM sites the Kleinsteiger lab has developed, we were able to generate cell lines with our mutation of interest. Now we're utilizing this expanded repertoire of Cas enzymes to develop corrective strategies for this mutation. To introduce this mutation, we utilize adenine base editing, which has an adenosine deaminase attached to the Cas9, so that adenine will be deaminated into inosine. This will be corrected to a guanine, resulting in an A to T to G to C base conversion. To correct this mutation, we're utilizing cytosine-based editing, which is very similar to ABE. However, Cas9 is bound to a cytidine deaminase. And so the cytosine you are interested in targeting will be deaminated into a uridine. This will then be read as a thymine, which results in a C to G to T to A base conversion. And so while we can model specific mutations, our proposed pipeline is to expedite this using NF1 patient blood. We would be able to take a sample of blood and sequence the genomic locus to determine the specific NF1 germline mutation of the patient. At the same time, from this blood, we can derive immortalized lymphoblastoid cell lines. While these cell lines are being developed, we can devise genome editing strategies that we can then test on our lymphoblastoid cell lines to determine the efficiency and fidelity of our corrective strategies. We would be able to optimize these strategies on these cell lines before utilizing some sort of delivery vehicle such as AAVs to then deliver our corrective strategy into the patient. Now, in the model I'm showing you, we are designating using this for neurofibromas, but it would work for any cell type. However, we have to continue our efforts to increase efficiency and fidelity of the CRISPR machinery in order to see these gene editing strategies reach the clinic. And so right now we have a collection of 29 LCLs that are derived from NF1 patients at MGH that we are going to be working on as a resource to test a number of different genome editing strategies. 2/3 of these LCLs are potentially correctable by base editing. And for those that cannot be corrected by base editing, we have prime editing as an alternative strategy, which we are looking forward to diving into deeper and seeing how this pans out. And so with that, I'd like to present my acknowledgments specifically to Peggy Wallace and Amit Hoke of the University of Florida and Johns Hopkins, respectively, for their cell lines, as well as the Children's Tumor Foundation, specifically Annette and Vidya, for all of their guidance in this project, the donors, patients, and their families, and of course to the Children's Tumor Foundation for funding this work. And with that, I'll take any questions. Thank you.
Thanks, Stephanie. So, yeah, we have the first question. I read it in case you haven't seen it. Have potential of target sgRNA binding site with this alternative PAM site been predicted?
Yeah, so there are tools, programs out there that you can use to look for off-targeted binding sites with these PAM sites. One of the biggest concerns with targeting NF1 is the fact that there are several areas that have pseudogenes. And so one of the things that we have been doing prior to working with these guides is to do searches throughout the genome to see, you know, is there any other area that they're going to target, and whether or not there is any residues within the area that we're looking to target that could be altered. And at least for what we've seen with the ones that we're currently working with, we see very few off-targeted effects.
Okay, while waiting for more questions, I have one. How do you think about, so when you can edit this mutation, how do you deliver it in the patient?
Yeah, so that's a really great question. It's one of those things that we, it's a work in progress, I would say, with the strategies that are available to get to the point where we could do that. Depending on what cell type you're trying to target, or what area, if you just want to focus on a plexiform versus some other type of cell type, you have to consider. So we have looked a little bit at AAVs. We've actually talked with Samantha Jin, who the previous speaker is from her lab, about trying to use some of their AAVs as a delivery mechanism. But there are a lot of considerations you have to take. And I would say that there's a great number of steps we have to sort of test out before we move into a patient.
Yeah, for sure. Well, there are not any more questions in the chat, so we can move to the next speaker.
Yes, so now we are delighted to welcome our last platform speaker from Spain, Nuria Catasus. Is a PhD student in the field of NF2, and she has been focused on the genomic analysis of NF2 tumors by exome sequencing, as well as in functional analysis to determine the pathogenicity of mutations in the NF2 gene and its implication in the patient's phenotype. And today she will present her recent work on antisense oligonucleotides as a therapeutic approach in NF2.
Today, I'm going to present a work that is titled Testing the Use of Antisense Oligonucleotides to Modulate the Effect of Splicing and Nonsense Genetic Variants Causing Neurofibromatosis Type 2. So, neurofibromatosis type 2 is an autosomal dominant disease caused by mutations in the NF2 gene. Its main feature is the development of bilateral vestibular schwannomas, although these patients may also develop tumors in other cranial or spinal nerves. It's a disease with a very variable clinical expression and for which a genotype-phenotype relationship has been well established. So it's known that the type of pathogenic variant is a factor that can determine the severity of the clinical manifestations. The therapeutic options currently available for patients with NF2 are surgery or radiation of the tumors. And there is also the treatment with bevacizumab that has been shown to improve hearing and reduce tumor growth in some patients with NF2. However, there is still the need to develop more therapies, and for this, the identification of clinically relevant and actionable therapeutic targets based on molecular analysis could constitute an opportunity. So it's largely known the relationship between the genotype and the phenotype of patients with NF2, in which missense or in-frame variants are generally associated with a milder phenotype of NF2 that presents later in life and with a slower growth of the possible tumors. In between, and with a more variable phenotype, we have splicing variants, which may depend on the position of the variant in the gene, among other factors. And finally, the truncating variants or large deletions are those that are associated with a severe phenotype of the disease, usually presenting at an early age with rapid tumor progression and usually with various additional schwannomas, meningiomas, or ependymomas. So in this project, we propose to test antisense therapy in vitro for NF2. There are different mechanisms of action for this type of therapy, but basically we can distinguish two main classes of antisense oligonucleotides. On the one hand, we have RNase H-dependent oligonucleotides, which induce the mRNA degradation. And in the other, we have molecules that produce steric blocking, which physically prevent or inhibit splicing progression. So for our purpose, we will be using morpholinos, which correspond to the last group, as they allow us the modulation of the splicing of the NF2 gene to modify the resulting form of medlin. In our laboratory, it has been previously studied an approach to use a morpholino in a patient with a deep intronic mutation in NF2. So this variant generated the inclusion of a cryptic exon, which resulted in a truncated protein. In this study, the effect of the variant was corrected at RNA level and showed medlin level restoration. So it demonstrates a potential use of these molecules as a therapy to correct splicing defects. So in this project, we propose two different approaches. On the one hand, we have the splicing variants, in which we use the morpholino to modulate the aberrant splicing signal produced by a point mutation. So we design a specific morpholino for each variant. In this way, the morpholino will be blocking this variant, and thus we will be able to recover the NF2 function and restore medlin levels. So we will be able to rescue the phenotype in vitro. On the other hand, for nonsense variants, morpholinos can be used to induce the skipping of the exon that carries the variant, as long as the skipping of this exon maintains the gene frame. Thus, and given the genotype-phenotype relationship, nonsense variants that cause a more severe phenotype could be modified to less harmful alterations. So first, regarding the approach of splicing variants, we have 4 patients to test morpholinos for whom we have designed a specific morpholino for each variant. And for nonsense variant, we have highlighted in blue the exons that when skipped, they keep the ring frame of the NF2 gene. So they are the exons that could be tested for this therapy. And in the laboratory, we have samples of patients who have truncating mutations in exons 4, 8, and 11. And we have designed a pair of morpholino for each of these exons, and we have tested these 3 exons. So just an overview to the experimental plan. From a point of NF2 patient, we establish a culture of fibroblast where the morpholinos will be tested. We set up the morpholino conditions at mRNA levels, and then we compare the treated and untreated fibroblasts to determine the effect of the treatment on Merlin and the in vitro phenotype. So we first tested the morpholinos for the splicing mutation. The first variant studied is a variant that causes the skipping of exon 15. When we look at the effect of the morpholino at the RNA level, what we see are the different isoforms of NF2, but we can see how the band that contains the skipping of exon 15 is the one that increases with the treatment. So we are generating the opposite effect as the morpholino is not recognizing specifically the allele which the variant with the variant and is altering the splicing of the wild type allele. The next variant studied induces the skipping of exon 8, and when we treat the cells, we see again the same effect as in the previous one, and we can see how it increases the band that contains the variant. So we tested different morpholino designs on these variants, and we see the same effect. And when we look at the protein level, we observe how Merlin levels do not recover. We next studied 2 patients with a variant near the donor splice site of exon 3. These variants produce the insertion of 7 and 10 intronic nucleotides, respectively. And once treated, what we detect is the skipping of exons 2 and 3. So when we use the morpholino to block these variants near the canonical splicing sites, the splice signaling is altered in a way that results in the skipping of the 2 exons. So when we look at the functional level, we see a reduction in Merlin levels. So in our experience, when the splicing variants are located near the splicing canonical sites, the treatment with morpholinos is not specific for the design variant and results in the skipping of the following exons. Thus, and in the case of NF2, this treatment does not allow to restore Merlin levels. The other approach that we tested is for patients with nonsense variants, in which the goal is to reduce the severity of the phenotype by creating a semi-functional form of Merlin, by inducing the skipping of the exon that carries the pathogenic variant. For these cases, we first set up the skipping of exons 4, 8, or 11 in control fibroblast. And once we know the conditions, we tested the fibroblast that contains the nonsense mutations and see what happens to the Merlin levels by Western blot. We show here that when we skip either exon 4 or exon 8, the Merlin levels decrease. Somehow here we would expect to see a smaller form of Merlin when we skip the exon, but the altered protein maybe is degraded, so we are not able to detect it. And finally, when we look at the levels of Merlin, when we induce the skipping of exon 11, what we see in this case are 2 Merlin forms, one smaller with the skipping of exon 11, and if we study the total Merlin levels, we detect a small improvement in the treated fibroblasts. We are now currently studying whether this form of Merlin without the exon 11 is functional and therefore could recover the phenotype in vitro. So the exon skipping approach to generate a semi-functional Merlin form to avoid a truncated protein does not seem to be appropriate for exons 4 and 8, since the use of these antisense molecules results in total Merlin loss. However, further studies need to be done to analyze the functionality of Merlin when skipping exon 11. Finally, I would like to thank all the people that have been involved in this project and all the funding agencies, and thank you all for your attention.
It's a wonderful talk. Are there any questions or comments from the audience? On Slido, maybe? Yes, so there is one question saying that the tumors with biggest need in NF2 are meningioma, especially when they are higher grade. They have quite a few other mutations. How does that affect your approach?
Well, in this case, we are only approaching for the NF2 mutations. We know that the meningiomas are quite difficult to approach because of these fewer more mutations. But in this case, I think it would be difficult to say what would happen with other. Maybe we could reduce severity or slow the growth, but I'm not sure. I think it's quite early to answer that.
Okay, and well, I have one question. After having demonstrating their efficiency, how would you deliver these oligonucleotides? I mean, do you plan a systemic delivery or intratumor delivery? How do you plan things?
This is quite difficult. Also, this approach has been shown to be useful in some disease from the retina, so that is easy to deliver because of this. It's like a kind of close, and it doesn't need to be systemic. And for Duchenne dystrophy is also be using, but I think that is systemic. I think it's quite. We have to prove that is useful. That is like could be useful, and in the in vitro models, and then we'll see how can we approach this. I think it's quite difficult, and it's early to know.
Thank you. And well, I have another question. Are there potential off-target effects using oligonucleotides? Or
yes, we know that these oligonucleotides have a 25 base pair, so they are supposed to be highly specific. But yes, we suppose that some off-target effects can be described, and if we demonstrate that it's useful and that it works, maybe we have to study further these off-target effects. Yes.
Okay. Thank you. Is there any additional questions from the audience?
Well, we have 5 minutes to discuss, and we have a general question for all of the speakers. So they are asking if there are any potential for activating NF1 pseudogene and then try to make them functional instead of correct the NF1 mutation. So if any speaker wants to give her opinion.
So the question is about influence therapy commercialization, or I hope I understand the question well. So what's
yeah, NF1 has a lot of pseudogenes, and like in other diseases, there are some approaches that instead of repair the gene that caused the mutation, is to try to do the pseudogene functional. So one of the audience asked if we can, instead of correct the NF1 mutation, try to express the pseudogene and then make it functional.
Yeah, certainly. I think Stephanie talked about that already. Use CRISPR approach. So my lab is also work on that. You can use CRISPR base repair or just typical HDR base repair. So you can correct all the mutations. So I think typically totally feasible. I think that. Of course, one thing relate to the mutation correction is that Stephanie mentioned there's so many mutations, right? I think you have to design, customize your therapy for specific patients. I think that typically is a major challenge. So another is that you can simply deliver gene replacement therapy. Of course, Stephanie also mentioned that the therapy doesn't exist actually at this moment because the gene is so huge, cannot be delivered. But actually, we show that the gene can be delivered using polymeric nanoparticles. So that's probably why we can see the amazing therapeutic efficacy in mice, in mouse models. So I think, so that's the gene replacement therapy could be generalized to most patients. But for mutation correction, it's certainly possible using CRISPR or oligo-based repair. So it's feasible.
Okay, so there are another question from the audience. I think it's this for Nuria. The effect of morpholino is only transient. So can you imagine a way to extend the duration of this effect?
Well, we have made this first approach, and we have studied the first 72 hours. I think that once we know that it works and that we can restore the phenotype in 72 hours, we have to evaluate which are the side effects for toxicity and see if we can extend, or we have to use more morpholino. But then again, it's like we have to take into account the toxicity. So I think that it's like trying to balance it, and it's difficult to extend. This is one of the contras of using these molecules.
Thanks, Nuria. So there are another question also, in general way. So how many years away we think we are for gene therapies to be delivered for patients? 10, 15 years?
Yeah, I think it's hard to know in a way, because the technologies are always changing. It seems like, we started out with CRISPR HDR, and then base editing came out. Now prime editing is coming out, and these technologies are constantly being updated and modified to improve them. And so I think there's still a lot of barriers we have to overcome. Like we were saying, NF1 is a huge gene, and right now we are looking at very personalized targeting of these mutations. If we were able to get to a point where, instead of worrying about a 20 base pair fragment, if we could get a whole exon, something like that, that would be more of like off the shelf CRISPR based editing. And so, yeah, I mean, I think in one way, I feel like it's closer than we think it is, and in another way, there's still a long way to go. But I to anybody else here, I'm not 100% sure how feasible 10 to 15 years is, but we've seen some CRISPR-based therapies already be put into patients in clinics, like with sickle cell anemia. So,
yeah, there are 2 clinical trials using CRISPR-Cas running nowadays. So, and your opinion, Dr. Zhou, about nanoparticles and restore NF1?
In the utility of nanoparticles. So, I think it's still at the early stage, right? You can see what I present today. We did see exciting results in animal models, but I think the good thing is that our particle platform is suitable for translation, and I think of course we need to further validate the findings. And then we're pretty optimistic about potential translation either for the gene replacement therapy. It could be the first gene replacement therapy for NF1, because the NF1 is huge. We can do that now, but also potentially for gene editing therapy. I know speakers here talking about using virus, and this is a standard approach for now. But I think use a CRISPR based repair, whatever. You can easily load it into the vector. So another thing to understand is that just like we receive Pfizer Moderna vaccine, and now you talk about new variants, right? You can just change the sequence and then load it into the vector again. So as long as the vector is proved to be safe, and then it's relatively easy to change the payload. So, for example, if our particles can be proved to be safe for patient use, then in the future, for example, we start with gene replacement therapy. A certain case is Stephanie or your therapy could be efficacious in vitro, even use virus. We can use we can load the payload into nanoparticles, right? We have the vehicle which have been proved. There's a potential promising routes there, but again, how to translate into clinic is remain a challenge, a major challenge, and it probably takes time. But of course, we hope we can move into clinic to help our patients as soon as possible. That's what we are doing at this moment.
Thank you, Oleg. So thank all of the speakers. I think it's time we move on. So thank all of you for your great talks and your participation. And for the audience, well, you have more or less 20 minutes to break. And let's see later in 20 minutes. Thank all of you.